Hope Lies in Dreams was a series of 10 podcasts that Brady did for Nature Biotechnology, which I think have proven to be very successful in that many people listen to them. And those podcasts focused on how one small biotechnology company, Ionis, that I established and led through all these decades, was able to create a new broadly enabling drug discovery technology, despite being really the only organization that pursued it for many years, and despite all of the negatives and naysayers who said it was impossible. And so, I mean, that is the focus. And then I suppose the secondary focus is on my life and how it led me to n-Lorem. It's about the challenges of doing something truly innovative, to the challenges of the extraordinary challenge of creating a new platform for drug discovery and development, which does not happen, and how it turned out that almost all of the progress happened at one organization called Ionis, and how and why we were able to do that, and why we weathered those many difficult decades or years to do what we did. First of all, how many drug discovery technologies are there? Well, the industry, it was founded on one small molecule drug discovery that started in about 1900 formally. And that is technology that continues to be the major technology that the industry uses to make drugs. Most of the drugs that you take are small molecules. That technology is extraordinarily inefficient, extremely difficult to learn lessons that you can apply from one drug to the next. And as a consequence, the number of innovative new medicines per unit investment has steadily declined. And so that has been the strength and the weakness of the industry. And the other new technology that is actually broadly enabling that occurred is monoclonal antibodies. And monoclonal antibodies were first identified in 1972. It took about 30 years, about 30 companies, many people in academia, and about $30 billion before monoclonal antibodies were developed and we came to understand what they could and couldn't do. And they are enabling, but certainly not as broadly enabling as small molecules. And at least initially, they were more efficient than small molecule drug discovery. In 1989, I founded Ionis to create a new drug discovery technology that's entirely different. All drug discovery programs before what we did focused on proteins, the molecules of the body that do the work of the cell. We didn't. We focused on the information management system of the cell, that is, how genetic information is transferred to create proteins, and that's called RNA. So it was entirely 100% different, an enormous change from, and the purpose of doing that for me, prior to Ionis, I was president of R&D at SKB, which is now GSK, a very large company. And before that, I built the first anti-cancer program in the industry at Bristol Myers. And prior to Ionis, I had been responsible for, I forget now, good number of drugs that are on the market, 15, 16. I took Tagamet over the counter, for example, and blah, blah, blah. I had very broad drug discovery and development, very senior experience in the industry. I was, if I remember right, 35 when I became president of R&D at SmithKline, and left because I felt that the industry was dying and it needed desperately an investment in a new technology that could do better for patients. And it was essentially considered impossible to even think about designing what are called ASOs, antisense oligonucleotides, to get into cells, bind to target RNAs, and do what they do. Everything had to be invented. It was a blank piece of paper. We had to invent medicinal chemistry. We had to understand how they actually get into cells, how they distribute, every single thing that needed to be done had to be created and then understood and then used to advance. And on top of that, of course, you have to make drugs to prove such an idea works. And not one drug doesn't do it. You have to do it over and over again. And as you know, drug discovery and development is 99% failure. And it's also extraordinarily time-consuming and costly. Average time to develop a drug after you've done the discovery today is 17 years, and the average cost is $3+ billion. Those are the numbers. So think about creating, one, the platform, and then a set of drugs that prove the platform works. And at the time I founded Ionis, there were another half dozen companies that were founded to pursue the technology. Only Ionis persisted, persevered, and succeeded. All the other companies either failed or moved on to do something that was easier. So we had to write a textbook, started from start to finish, and we did. And that spawned now, scores of companies that are pursuing the technology. And, a number of drugs that are on the market. And at last count, probably 100 to 120 drugs that are in development around the world. And the technology has proven to be more efficient. It also is much more specific. And so we can do things that other technologies can do. And there are very few undruggable targets for us in contrast to other technologies. So it's an enormous undertaking. It was an enormous challenge. And we had many, many disappointments and failures along the way, and many setbacks. Well, that's different. Discovering drugs and developing drugs is a derivative of a technology. Creating a technology that will allow you to discover many, many drugs is a very different kind of challenge. Mm-hmm. And it's damned hard. It takes a long time. It has to be associated with failures and disappointments. And the only way to do it is to have, a plan that focuses on innovation over the long haul and committing to the technology and solving problems one at a time, addressing the issues, and then moving on. And you have to do it realistically. You have to recognize that most of the time these ideas fail. And so you have to be prepared to commit to the dream, but also prepared to acknowledge that it doesn't work if you get to the place where you see no path forward. And we faced that on many times. And each time I did, I sat down and looked at all the data and felt that we had a path, and we announced that. And as a general rule, it was greeted with a great deal of skepticism. And skepticism is appropriate. That's the heart of science. And anybody who sets out to do something like we did, everyone should be skeptical. And so that didn't bother us. But so those are big lessons in that you have to create a special environment where innovation and innovators are the center of the of that, and you have to create an environment that will do that over decades and decades. It's one thing to be an innovator in high tech where an idea can be converted to a product in a year or two or whatever. We can't do that in our industry, and that's why it's so much more difficult. So it's not just money, and it's not just failure and disappointment and challenge. It's also time. Yeah, my goals in founding Ionis were to create, if possible, this technology, validate it, advance it, and make as many good drugs for patients as possible. I'm motivated entirely by the needs of patients. That's the only thing that drives me. Business is just a means to pay the bills. That's probably why our stock never got to 200. But in the end, it's about the patient. And I also set out to create a unique culture that was hyper demanding, but supportive of individuals, that focused on creating an environment where innovation was the center, and that innovators were given the space to be innovators, which is not an easy task. Innovators generally are pretty difficult to live with. Having been an innovator all my life, I know that I've been difficult to live with on occasion. Don't you laugh, Amy? Where the central reason to come to work every day was sick people dependent on us, and where the only drive was to do basic science and to advance the technology, and to build that and to maintain it over many decades was a challenge, and I think to a very real extent we did that. Now I'm proud of that, and without the culture, without the plan, without the commitment, this technology wouldn't exist. And so, it's interesting. One of the major journals has asked me recently to write a perspective on how I created the culture that led to such an rigorous science, rigorous science over the long term, and integrated innovation is the only way we could succeed. And so that article is coming out in the next couple 3 weeks. And again, I think that's very flattering of the organization that I was asked to do that. But again, I think everything is integrated, right? I mean, what I did, since our task was unique, I created a bespoke culture. I designed everything from scratch using all the experience I had in large companies, but it was geared to doing the tasks that we had for ourselves. And where I'd be asked to do a different kind of task, I wouldn't build that kind of culture, but it was that kind of culture that was required for us to be successful. And I think it's fostered growth in many, many people and led to a lot of great science and some great medicines, and it's going to lead to many more, which is something that we should all be happy about. I do indeed, and but it also means, it's not it's not right for everyone. It was very clear. I made it very clear at Ionis that Ionis isn't right for everybody, and it became very clear over time that people sort of divided into those who left after 2 or 3 years and those who stayed. And since we were creating new knowledge, one of my major goals was to retain long service employees. And For many decades, well, for many years, our turnover was less than 4%. Biotech turnover on average was 26%. And of the original 30, I think about 15 are still at the company or have retired from the company. I'm really proud of that. And again, I think the goal was not to create an environment that everyone would thrive in. The goal was to create an environment where the people I wanted, would thrive. And it did that. And in that sense, then it's transferable. What the systems I put in place to foster rigorous science are, I think, very transposable. And those are all sort of outlined in this perspective that I've just written on how I did it. So yes, but transposable only to a certain set of goals and to a certain kind of leader and a certain kind of people. The environment was very interrogative and is. Science is an interrogative process. It's never done until peer reviewed. And as a scientist, I'm very comfortable with interrogative interactions and very comfortable with ambiguity and probabilistic answers. Scientists never give definitive answers, right? That's not the case for many people. Business people, especially, are really uncomfortable with all that. So it is transposable for the right reason with the right people and the right leadership. And it worked. It really worked. The neat thing about RNA-targeted drug discovery is still advancing at a tremendous pace. And it will continue to do that for a while, which makes it much more exciting to me, because that means the performance of drugs and the things we can do will continue to advance and more patients will benefit. So, and as you know, now people are, designing small molecules to do it. And again, that's just using small molecules for what we've used them for 100 plus years. But that doesn't make it unimportant. It's just a new application. So, the things that I'm excited about looking forward as a person really more interested in creating the sort of base from which multiple drugs can come are some of the new approaches that really don't look back at monoclonals or look back at that sort of thing, but look forward toward new approaches that I think will add value.